with some of you. So, you know, when I was growing up in Southern California, it was pretty much assumed that I was going to go to college. I was on a college preparatory track and everything was all fine and dandy. And then the middle of my senior year of high school, my mom got cancer and she passed away. And I was really, you know, lost and confused, and I was a first generation college student, and so my dad was an airline pilot who came out of the military. And so I was really sort of on my own of trying to figure out what I was gonna do and how I was gonna go to college. And because of, even though we had, you know, good health insurance, it's such a classic American story, even though my dad had good health insurance with the airline, you know, my college fund pretty much got eaten up by the part that the insurance didn't cover when my mom got sick. And so instead of, you know, going right to a four-year university, I went to a community college. In fact, I went to 6 different community colleges, because in that summer before my senior year, when my mom was in the hospital, I met with my high school counselor, and at the time I was really involved in, you know, science and math. I was in honors and AP and all that kind of stuff, but I was a theater nerd, and I didn't really know what I wanted to do. I kind of wanted to go to UCLA with all my friends and be a theater major, but I also really had this love of marine biology. And so, you know, I went to have this conversation with my high school counselor, and he said, "Oh, honey, girls don't do science." Not even kidding. In 1981, girls don't do science, stick with theater. And what did I know? I was like, oh, all right. And then long story short, through a series of moves and, you know, other things that happened after my mom passed away, I went to 6 different community colleges in like 3 different states, trying to figure out what the heck I wanted to do. And so I finally ended up at a community college that just happened to have marine biology as a class. And at that point I was just racking up general education credits. And so I knew I was gonna have to have a lab science. And so I took marine biology, and I was like, oh yeah, that's what I wanted to do. And I was 22, 21 by the time that happened. And so, you know, at that point I was able to focus on what I wanted to do, and I got my associate's degree, and I transferred to UC San Diego. But I had more bad advising because, again, first generation college student, didn't know what was going on. And I ended up at UC San Diego thinking that I could go to Scripps and be a marine biology major, not knowing that at the time that was only a graduate program. They have an undergraduate program now. So I ended up being an anthropology major because I knew I wanted to study animal behavior, and I happened to get lucky and meet a faculty member in anthropology who studied primates and dolphins. He was an expert in cognition. And so, you know, through a very circuitous route, I found my way. And part of what helped me to succeed was just being tenacious and just saying, you know, okay, well, that didn't work the way I thought it was going to, so I'm gonna just shift gears and, you know, try something else. And just not giving up on what I knew I wanted to do. And then once I graduated from college, you know, I had a little bit more self-awareness, I had a little bit more knowledge about the system, I had a few more tools to help me figure out how to navigate, you know, all of this stuff. And I figured out how to go and talk to my professors about what I wanted to do. And so then my trajectory from there was pretty straightforward. So I went to San Francisco State and I got a master's in physiology and behavioral biology. And that was the point at which I realized, okay, I think I like the physiology side of things more than the behavior. So that shifted my plan. And then I went to Texas A&M University for my PhD in wildlife ecology. And I did my doctoral research up in Alaska. And I worked on stellar sea lions. And then I stayed in Alaska for a little while and did a postdoctoral fellowship in Fairbanks, but I did my research down in the Antarctic. So it was like top to bottom as a postdoc. I used to make a really inappropriate joke about being bipolar as a postdoc, and then I realized that was not really a good joke, because that's not a funny thing. It's a very serious thing. So, and then, my faculty life, I was at a small private university in Anchorage called Alaska Pacific University, which is why you see that logo. Because when I became a faculty member, I started my own, lab. I was the only person at the university studying marine mammal physiology. And so I created the Marine Physiological Ecology Laboratory. And actually one of my undergraduate students drew the logo for me, which was super cool. And I got to pay her to do that. So that was really fun. So that logo is up here because the work that I'm going to tell you about today, I did when I was up in Alaska. So it's not really connected to my life at Eastern. So I was a faculty member for almost 15 years, became a department chair, realized I was really good at administrative stuff and really liked it, and found that I could reach a lot more students as an associate dean and eventually a dean, and who knows what will happen after that, because I can do big university-wide initiatives in a way that I couldn't when I was just doing pure research. So people ask me all the time, well, don't you miss being in the field? And I do. I miss, having a blue wellhead in my lap. That's a pretty fun thing to do. But I really love being at a regional comprehensive university where I can connect with a really wide community of students. So that's a little bit about me, and really, my main message for that is, be tenacious, don't give up, don't be afraid to change course if things aren't going the way you think they should, or if you decide your interests change, and don't let anybody tell you you can't. So those are kind of my big takeaways. So, but let's talk a little bit about science and about marine ecology and the kinds of things that you can do and some of the things that I've done as a marine ecologist. So, marine mammals are a really interesting collection of species because they are mammals that live in the aquatic medium. Right, so they're mammals like us, so we breathe through lungs and a cardiovascular respiratory system. We breathe air, so not like fish, no gills on a whale. And so the challenges of living in an aquatic medium are very specific, and marine mammals have very specific adaptations that allow them to live in an aquatic medium. And I'm going to go through some of these next couple slides really quickly because it's not important to me that you really understand the science or think about the science per se, but this is an example of why, as somebody who wants to go work with marine mammals or other animals out in a marine system, you need to know your physics. Because physics controls a lot of how marine mammals live in an aquatic medium. So seawater is 800 times more dense than air and 60 times more viscous. So when you are a mammal trying to navigate making a living in an aquatic environment, you have some additional challenges that are placed on you by the environment that you have to adapt to. And two of the most important ones for marine mammals in particular that are related to the whole question of where's the fat are buoyancy, how do they stay afloat basically, and how do they control their position in the water column, and drag. Right? So how do they swim efficiently? Think about, if you're in a swimming pool, if you're any of you swimmers or triathletes in here, right? So you know that you have to be thoughtful about your body position in the water in order to be streamlined and minimize drag, right? And think about if you have a body shape that's not designed to be swimming through the water, or let's say, if you're swimming with a backpack on, right? That's going to make it so that you are not efficient in the water. So buoyancy and drag are two really important things for marine mammals to make sure that they can optimize their energy. So again, the science here is not necessarily important for you today, but thinking about the things that determine buoyancy for an animal, right? Their body mass, how much they weigh, the acceleration, which in this case is really the force of gravity, and the density of the medium, and then the volume of the body that's in water. And so because buoyancy has this specific equation that drives it, in order to avoid sinking as a marine mammal in an aquatic medium, there's only a few things that you can do. And the easiest thing to do is increase the volume so that you're less dense than water. So air sacs, right, your lungs, the lungs of a marine mammal can increase the volume of the body, but so can fat. So we're going to talk a lot about blubber today. Same with drag, right? How an animal manages the flow of water across the body's surface and tries to minimize drag is going to relate to buoyancy, how you can be afloat in the water, but also how you move through the water in an energetically efficient way. And if you think about where you swim, right? Any scuba divers? Okay. When you're swimming in scuba gear, right? If you're going from your dive site back to the boat, are you swimming at the surface or are you swimming under the surface?
Under most times.
Most of the time you're swimming under the surface, right? Because the coefficient of drag, so the force of drag as a function of speed, right? You can be submerged and you can have a lot less drag as a function of speed than when you're at the surface, right? Because there's basically, and it's been a long time since I've given these lectures. There's a point at which you go down in the water column, and it's about a couple of meters down in the water column, you know the metric system, a couple of meters down in the water column where drag becomes significantly reduced because there's a certain amount of drag that's just created by the surface of the water, right? So when you're submerged, your drag is a lot less as a function of your speed and how you are creating flow around the body. So, and then we can think about types of flow, right? Again, physics. Pay attention in physics, right? If you have a nice smooth body moving through the water, what you're trying to do is maximize laminar flow, right? So that the water is moving smoothly across the surface of your body, and minimize turbulent flow, which creates frictional drag, right? And reduces your efficiency. And so depending on, how the shape of the body, as the water moves, as you're moving, creating a space in the water as you're moving through, you can either have laminar flow across the body or turbulent flow. And every time there's a structure that sticks up in the water, like a dorsal fin, a scuba tank, right, that creates turbulent flow. And we don't want turbulent flow. So those are things to think about as we think about how animals move through the body, and we're going to get to the importance of fat part really soon. Other ways that you can minimize drag for these types of animals is the size and shape of the appendicular, as in appendages, or axial skeleton, right? So some of the adaptations, anatomical adaptations of marine mammals for being in that aquatic medium have to do with how they have evolved the shape and position of the spine relative to the rib cage, relative to the appendages, and the relative shapes and sizes of those parts of the skeleton. So they have all the same type of parts of the skeleton as all mammals, right? We can identify the same pretty much individual bones. Some of them have gone away. So this is the pelvis right here. Whales still have a pelvis, but it's greatly reduced and it just kind of floats around in their torso as a vestigial part of the skeleton because they don't need it anymore, right? Because the hind limbs have evolved into their tail flukes. So that helps to minimize drag as well. But the most important physical component of a marine mammal that minimizes drag is blubber. Because blubber, which is just fat, blubber helps to smooth the body contours and reduce turbulent flow across the body. So even when you have things like dorsal fins and pectoral flippers, you can still minimize that turbulent flow by having a layer of blubber across the surface that smooths out those body contours. So blubber has a very specific structural purpose in helping with buoyancy and also with minimizing drag. So let's talk about some other things that make fat really important. And this is gonna get to sort of the meat of the talk, and I'll show you some research. So we've talked about streamlining for hydrodynamic swimming. Blubber is also obviously an insulation for marine mammals that keeps them warm in cold water. You lose heat to water about 75 times faster than you lose heat to air. It also provides physical structure for protection of internal organs, and for pregnant females, it also protects the fetus. And it's an indicator of body condition. So some of the things that we're going to talk about today are how we think about blubber in terms of understanding whether or not animals are healthy and potentially experiencing things like nutritional stress. And particularly for marine mammals in cold climates like Alaska, the Arctic, the Antarctic, and for species whose primary prey, their food, is really abundant in the summer, but not abundant at all in the winter. So very often they're fasting in the winter. Some species also fast during part of the reproductive cycle. That blubber is an energy store that can be mobilized or metabolized. So if you look at, this is a cross-section of blubber from a bottlenose dolphin, but it's pretty emblematic of most species, particularly species that have seasonal fasting in their ecology. So if you look at the blubber, so the epidermis, the skin layer is clear there, and you can see some of these gradations in color. And if you were to really look at a piece of blubber, I wish I could have brought a hunk of blubber. If you really look at a piece of blubber, you can see structural differences in these layers. So the outer layer primarily provides that structural support, the smoothing of the body contours, and is fairly dense, and it's not well innervated, or it doesn't have a lot of nerve endings. The middle layer is more of that thermal protection layer, and then the inner layer that's connected to the muscle is that metabolically active layer that they can strategically pull in those blubber resources to get them through a fasting period. And if you analyze the fatty acid composition of the different layers of blubber, all the different layers have different proportions of polyunsaturated, monounsaturated, saturated fatty acids, depending on whether it's a metabolically active layer or a structural or insulation layer. So it's really, really interesting how highly adapted this one tissue is, which is basically just fat, for these animals to be able to survive and make a living in that aquatic environment. So some of the research that I did when I was in Alaska was on beluga whales. Delphinapterus is the genus. And there are 5 different populations in Alaska. And what you'll notice about this one population here in Cook Inlet, what looks different about this population than the other 4 populations, just sort of right off the bat? Can you see something that looks unique about that population, just from the geography?
It's a lot more, it reaches a lot more inland.
Okay, it's more inland. What else?
It's pretty small.
Small. Mm-hmm. What else? Think about the geography part.
Pacific Ocean.
Okay, it's southernmost. What was the other?
I said like the Pacific Ocean versus up in the Arctic.
If I were a beluga whale in, I don't know, the Beaufort Sea, could I go hang out with my buddies in Cook Inlet?
It's pretty isolated.
It's isolated. Okay, so that's really, really key. So Cook Inlet beluga whales are genetically distinct because they are geographically isolated. So unlike all of the other 4 populations where there is some mixing, right? So when we look at the genetics of the other 4 populations, you can't really tell them apart in a significant way. I mean, there are markers for Bristol Bay, the Bering Sea, and so forth. These 2 populations mix a fair bit. The Chukchi and Beaufort Sea populations mix. They're also mixed with populations in Canada and Russia. But Cook Inlet is completely geographically isolated from all other populations of beluga whales. And so it is so genetically distinct that it's very close to being listed as a separate subspecies. So they're non-migratory, obviously, they're kind of stuck right where they are. And they were listed as endangered under the Endangered Species Act of the United States as in 2008. And they had critical habitat designated in 2011. And so if you look at, this is sort of the general distribution of Cook Inlet beluga whales. So this is that little call out part from the other map. This population being critically endangered, their entire home range is in the most industrialized part of the entire state of Alaska. And there's a major military base there, and the military base has an exclusion zone that is pretty much the most important part of their critical habitat, which is sort of right in this area right here. So acreage is kind of right here. So they're really kind of stuck where they are. Part of the reason that they are as endangered as they are was unregulated sport hunting. A lot of people don't even know about this. So one of the interesting things that happened with Cook Inlet beluga whales was that when we realized that they were endangered from doing these population abundance estimates, back in the early 90s, everybody assumed it was the Alaska Native subsistence hunt that was the culprit, because these are a very important subsistence resource for Alaska Natives in all of the populations in Alaska. But actually, as late as the 1970s, early 1970s, when the Marine Mammal Protection Act went into effect in the United States that prohibited non-subsistence hunting of any marine mammals in the United States, there was a big game hunt, a sport hunt for beluga whales in Cook Inlet. And I have a whole separate talk that talks about the history of hunting of Cook Inlet beluga whales, but they're like the Stay Puft marshmallow marine mammal. Their skin is like this buttery soft leather, and it's snow white in adult animals. And so they were mostly hunted for their hides to make luxury goods. And so we didn't actually realize how far the population had decreased until about the 90s. We think the carrying capacity for this population in Cook Inlet is about 1,200 animals. And by the early 1990s, there were maybe 600 or 700 animals in the population. How many of you are interested in math and statistics? Okay, so one of the things that you'll notice, so this is the population abundance over time of beluga whales in Cook Inlet. And one of the things that you'll notice, particularly as we started refining our models for how we estimate abundance, which basically you do aerial surveys and you try to count these white blobs as they pop up in the water. So it's very hard to count whales because they spend most of their time underwater where you can't see them. And so you have to develop correction factors, mathematical correction factors for refining your abundance estimates. So one of the things that you'll notice over time is that the error bars on these abundance estimates, back in the early 90s, were really might have been 400 animals, might have been 1,000 animals, we don't really know. So this is a mean estimate, but over time, as we refine the correction factors on our models, those abundance estimates get much, much better. And I haven't updated this slide in a while, I apologize. There's about 250 animals left now as of the last survey in 2018. In 1999, and part of the reason that this stabilizes a little bit, in 1999, the Alaska Native tribes around the Cook Inlet region instituted a voluntary moratorium on subsistence hunting in order to give the population an opportunity to recover. They have not recovered. Beluga whales produce only one calf about every 2 to 3 years. They stay with their calves for quite a long time. And so if you think about there being 250 animals, you assume half of those are females. You assume a third of those are having babies in any given year. And then you figure just even natural mortality rates for a marine mammal calf is somewhere between 40 and 50%. That's not very many new animals in the population in any given year. And so even with a moratorium on subsistence hunting and the Marine Mammal Protection Act coming in and their ESA designation, they're still declining at about 1 to 2% per year, and we don't know why. So, and the other thing that has happened that has made it really difficult for these animals is they've had what's called a range contraction. So as the population gets smaller in number, they tend to hang out in smaller and smaller and smaller portions of their habitat. And there are a variety of reasons for that. But the thing that makes it difficult for Cook Inlet beluga whales is that because they primarily eat salmon and these little fish called hooligan that are kind of like herring or capelin or sardines, and the source of those food sources is high up in the inlet from all of these river sheds that come down and feed Cook Inlet, their range contraction has contracted them into the worst part of their habitat in terms of noise and coastal zone development, oil and gas exploration using sound, construction of one of the infamous bridges to nowhere. So they've constricted themselves into the most difficult part of their habitat to live in. So this brings me to the actual research part. So there's a large team of folks who got together as part of the recovery plan for the Cook Inlet beluga whales to try to understand what does a healthy beluga whale even look like, right? What kinds of blood parameters does a healthy beluga whale have? What kinds of body size and shape should we expect in a healthy beluga whale? And then I was also, my personal area of expertise is in the energetics and physiology that drives behavior. And so I was really interested in, what is their blubber condition in a healthy beluga whale? What does that look like? So that when they're not feeding over the winter because there's no salmon in the winter, how are they mobilizing those blubber stores to get them through the winter? And what does a healthy population of beluga whales look like in terms of their blubber morphometrics, the physical aspects of their blubber, as well as the energetics of their blubber? What's the proportion of fatty acids and those kinds of things in their blubber? And so we wanted to establish a baseline of a healthy population so that we could eventually compare that to animals in Cook Inlet. So we studied the population in Bristol Bay, which was that closest kind of next door neighbor population around the other side of where the Aleutian Island chain comes off on that map. And the population that's in Bristol Bay, Alaska, is a healthy and growing population despite a very active but well-regulated subsistence hunt. And so we partnered with the Alaska Native hunters to do these health assessments on beluga whales in Bristol Bay to establish this baseline. And so we would go out with the hunters. They would help us wrangle a whale, basically in a skiff. We had some folks from Georgia Aquarium and Mystic Aquarium who were experts in, passive restraint, so that we could basically capture a single animal, put it in this nice, you can see it's kind of in a sling here on the sides. You kind of pull it up into shallow water so that you can work with the animal, but it's still, you know, in the water essentially. And then we could basically give it a physical. So we took blood, we took more, you know, measurements of girth and things like that. We took skin samples. We would have them, you know, blow from their, when they would breathe from their blowhole, we would put a little petri dish over the blowhole so that we could see what kinds of microbiome things were going on. So, you know, took fecal samples, all of it. So my part, of course, was looking at the blubber. And so there's a couple of different ways that you can look at blubber structure in a marine mammal, right? You can take it out of a dead animal. You can take a biopsy, which is basically punching a giant hole in the animal and taking out, you know, a core of blubber, which is really invasive. It's actually not that painful for the animal because the skin and the blubber don't have a lot of nerve endings in them, but it's invasive and it has the potential for, you know, infection and those kinds of things. Plus they're protected, and so it's really hard to get a permit to do that kind of thing. And so there have been a few studies on pinnipeds, on seals and sea lions, using high resolution ultrasound to look at blubber. And also, blubber distribution is different in different parts of the body. So if you're only taking one biopsy, you're only getting a snapshot of part of the animal that may not reflect what's going on, you know, from head to tail. And so this project was the first time that high resolution ultrasound was ever used on a beluga whale, so pretty happy about that. It was one of my favorite publications. So basically, as we captured the beluga whales, so, you know, you can see the little ultrasound tool here, and there's a person, you can't see in the picture, you can see the hat. There's a person with the little portable ultrasound machine who's guiding, capturing the images. And so we took ultrasound images from 10 positions across the body, on the dorsal side, the lateral side, and then right here under their front pectoral flipper. Because from previous studies of stranded animals, we knew that that was a really important location for blubber depth. And so this is why you need, well, that's the next slide, but so this is a picture of what an ultrasound of blubber looks like in a marine mammal. And so you can kind of see, I'm a pointer, but you can kind of see at the top there where the A mark is. At the bottom of that A mark, that's that epidermal layer, right? That's the skin layer, and their skin is really thick. And then you can see as you go down the layers of blubber to, this is the blubber muscle interface right here, how the structure and density of the blubber changes, right? The higher density of the blubber, the brighter the white, right? And so, we were able to measure the depth of the blubber, and then in a separate study, one of my other graduate students looked at the energetics of blubber, what's the caloric content of the different layers and so forth, for using it for a food source, basically. Because this had never been done in beluga whales before, we needed to calibrate the measurements for the ultrasound. Were we measuring to the right place in that ultrasound photo? And so we went up to the Chukchi Sea population, where they were subsistence hunting, a population of beluga whales up there. And we were able to get access to some blubber samples from freshly hunted animals so that we could compare. And we did a double-blind comparison, so the people who were measuring the blubber here did not have access to the ultrasound images. So we took ultrasound images on the carcasses before we harvested the blubber, and then we harvested the blubber and measured it. This is where you need to take statistics, right? So we looked at, for each individual sample, the ultrasound depth and the ruler measurement depth, to make sure that we were had a strong matchup of our measurements, right? So this is basically a general linear regression, and when you have your R squared value, the closer your R squared value is to 1, basically means that you did a really good job of predicting. Could you predict the depth with a ruler by taking an ultrasound, right? So our R squared value was 0.87, that never happens. When I saw that analysis, I was just like, yeah! Because we knew that we were looking at the image correctly and measuring to the right place in the ultrasound image. The take-home message from this, right? So we had different age classes of animals. This is basically tells you a couple of things. So we measured adults and juveniles. We measured animals in the spring, right after they've had to go through a winter where there's, mobilizing those blubber stores, and we measured them in the fall when they're fat and happy on salmon. So all you need to take from this is that, so these are the measurements from all of those different locations on the body. You can see how variable the blubber depth is across the body, right? In some places it's really thick, and in some places it's really thin. And so that just kind of confirms for us that you can't just take a biopsy and understand what's going on with blubber in these animals, right? So in order to understand sort of the overall body condition picture for these animals and understand sort of get kind of a generalized total body fat, right? What are their total blubber stores, right? How much do they have available that they can use? We had to figure out how to estimate the mass of blubber relative to the total body mass of the animal. And normally this slide takes me about 10 minutes to go through just on its own, but I'm going to give you just some take-home messages. So we had a lot of information from the literature from harvested animals about, generally speaking, what proportion of a healthy beluga whale is blubber. And so that was a study by Sargent and Brody all the way back to 1969. So it's really important when you're doing science and you're doing a literature search, you can't just look at stuff that's been done in the last 10 years, right? There are seminal papers that you need to make sure you do your due diligence and go back and fully understand all of the literature that's relative to the thing that you're trying to study. So generally speaking, a healthy beluga whale has about 40% of its body mass is blubber. So we use that as sort of our generalized model. But we didn't want to just rely on that. We really wanted to understand exactly what was going on with each individual animal. And rather than just say, okay, we're going to assume it's 40%, we're going to see if our measurements on these live animals actually come out the way that Sargent and Brody thinks that they do, right? So part of the reason that we did all of these different locations around the body was that Gales and Burton in 1987 came up with a model for elephant seals that uses geometry. Yay, more math! So you basically turn your animal into a cone and you use basic